Welcome back, everyone. Hope you guys had a quick break. So now we are back for more exciting talks. Uh, for our next presentation is going to be by Maílis Carrillo. So they're going to talk about influence of caging conditions on the behavior, physiology, and post-release success of wild wood mice. So thank you again for joining us today, Maílis, and you can start at your convenience.
Are you seeing my presentation?
Yes, we can see your presentation.
Okay, perfect. So, my name is Maílis and I'm here today to present my work entitled Wildwood Mice in Captivity, Effects on Behavior and Physiology and Post-Release Success, which is a part of my ongoing PhD thesis. So, there are many animals kept in captivity for research. Among those animals, there are two different types of models that are usually involved. So the laboratory animals breeding for that purpose and wild animals, which of them being more suitable for certain studies. So the use of animals for experimentation in EU is regulated by the Directive 2010/63 EU with the aim of standardizing the conditions under which animals are kept in captivity. It is specially focused on laboratory animals and addresses a wide variety of topics such as ethics, care and accommodation of animals and killing procedures in studies with the terminal endpoint. So this directive is also applied to wild animals. However, the guidelines for this type of animals are few and not very specific. There are only vague recommendations about the transport, release, and permits needed for studies involving those animals. So, and what is the problem that comes from this? Is that the fact that laboratory animals are very different from wild animals in several aspects, for example, their physiology and behavior. So, one of these differences is that wild animals show a period of adjustment when kept in captivity. In a previous study, we already confirmed the adjustment to lab conditions of wild wood mice. And why we use the wood mice is because it is easily found in wild and often used in research, as you can see in several studies. So our results on this previous study showed that the lab condition occur mainly, that the adjustment to the lab conditions occur mainly physiologically, and that there was a complex interplay between behavior and physiological adjustments. So, considering that is already known about the adjustment of wild wood mice to captivity, we decided to manipulate the animals' keeping conditions. So we know that animals are mostly kept in standard laboratory conditions. On the other hand, it's already known that natural enrichment is beneficial for animals. So in this context, we thought that it would be interesting to see if more natural enrichment could change the animal's risk avoidance behavior, and even if their survival after release can be affected by this. Thus, the aim of this study was to understand if and how different maintenance conditions could alter the behavior of animals in captivity and their success after release. So for this study, we followed a field lab field approach in which animals were captured, kept in an animal facility, and released and recaptured for monitoring. So, for that, the animals were divided into three distinct groups, one composed of animals captured and immediately released in their natural habitat, the field group, and the other two kept in the animal facility. The two groups kept in the animal facility were divided between standard, the lab group, and more natural maintenance conditions, the net group, both on the enrichment provided as well as on their diets. So, in the animal facility, animals spent 30 days being involved in non-invasive experiments once per week. So before starting the weekly behavioral experiments, the body mass of all individuals were registered. Regarding behavior, we observed the development of stereotypic behavior such as bar mounting, backflipping or patterned running to evaluate the animal's welfare. A light dart test was also performed to collect data on the animal risk avoidance behavior, such as the number of crossings between the different sides of the arena and the time spent on each side. Additionally, data regarding urine marking patterns was collected to assess the animal's exploratory and or dominance patterns. So, all animals were marked both in the field and in captivity with P tags. Soon after release, animals' recapture sessions began, lasting a total of 4 weeks with 3 days per week. So, to analyze the obtained data on behavior and physiology, we made models which the response variables of our interest, in which we add as fixed factors the caging conditions, the week of testing and sex, and additionally the animal ID as random factor. As the response variables, there are the parameters that we consider to be relevant, such as the body mass, the number of crossings performed in the light dark test, the proportion of time spent performing stereotypic behavior, and the patterns of urine marking. To analyze the ecological data to assess the post-release success of the animals, we used a well-known capture mark recapture model, the POPAN, to understand the dependence of experimental groups, time, sex, and weather conditions on the probabilities of survival and capture of those animals after release. So, starting with our results, we verified that males were heavier than females in captivity, which we associate to the sexual dimorphism of wood mice that is already known, with males being bigger than females in general. So, regarding the results of light dart tests, we observed that animals kept in more natural conditions performed more crossings between the light and dark side of the arena, especially in the first week. Additionally, we verified that the average of these crossings decreased over time. Considering those results, it concluded that more natural enrichment can delay the decrease in activity but does not prevent it from happening. So, regarding the stereotypic behavior, we did not find any significant differences. We assumed that it might be related to the fact that lab conditions had enough enrichment provided for the animals. Nesting material and shelter material were also constantly present in lab group cages, and those elements are known to be essential for the maintenance of animals' welfare. And it might explain the absence of differences between those groups. So, regarding the urine marking, we found that animals in lab conditions marked more the marginal area of the cage, especially in the first week, and that the marking decreased along the captivity period. The sex difference did not seem to be very informative due to its high variability. Thus, we think that the explanation to these results is related to the fact that animals in lab conditions have a higher scarcity of natural cues, leading to a higher need to mark the area. Additionally, we think that the adjustment to captivity, which was already observed by us in our previous study, might explain the decrease on marking patterns along the time. So, regarding the success after release, we found that the probability of survival and capture depended on time and weather conditions, respectively. In other words, the treatment to which the animals were exposed, so the net and the lab treatment in the animal facility, did not affect the success after release. As such, we conclude that the captivity has no influence on subsequent success of the animals. As a conclusion of this study, we observed that more natural conditions delay but do not prevent the adjustment to captivity. However, the use of these maintenance conditions is recommended for short-term studies that do not exceed 1 week to obtain more reliable results. Furthermore, we found that captivity does not affect the success of animals after release, and it leads to our take-home message that the maintenance conditions of wildwood mice in captivity do not compromise their readaptation to natural habitat. So thank you all for being here and listening to what I have to say, and to entertain audience a bit more, I brought for you a cute video of a wood wild mice being released. And thank you.
Well, that was a really interesting talk and interesting results because we do a lot of experiments with especially mice and stuff in captivity. Uh, so there is already a question. So question asked by Tang. So can we avoid recapture? Did weather conditions influence survival probabilities?
Sorry, I didn't understand the question.
Uh, can we avoid recapture? Did weather conditions influence survival probabilities?
So if we can avoid the recapture,
Yes.
okay, I understand. So weather conditions can affect the survival of the animals because so what we saw is that in certain times, for example, in a hot wave, we saw that many animals are not recaptured. So we don't know if it's exactly they can be there, actually they can be there, but we are not able to recapture them. So it's because of that that weather conditions influence the probability of survival.
Um, so what about the mice that are born in the lab? So can you, if you put like naturalistic conditions, immediately after they were born, for example, is that
Okay.
going to differ their behaviors and all the other parameters that you mentioned?
So we avoid to have pregnant ladies in the lab. So in the capture of the mice, we search for signs of pregnancy and try to not bring to the lab the pregnant lady. So actually, it happened once. To have babies in the lab. So what we did is to stop all the experiments for this mouse and to provide many more enrichment for the animal. And after we feel that the animal and the babies are enough with enough development to be released, they were released with the mother.
So we have a question in the YouTube chat.
Yeah.
Ellen asked, I am curious about the urine markings. Are they used to recognize the environment?
So yes, actually, although the wood mice are solitary mouse, a mouse species, they have the habit to mark the environment. So it's because of that that we think the absence of natural cues in the lab conditions make them to mark more on the first week than the other ones that have all the things, all the enrichment of the natural enrichment, that don't have this need to mark the territory.
Um, Another question on YouTube by Tang. Were there differences in the rate of weight gain across different caging conditions?
No, actually, we didn't found differences on the gain of weight, which is interesting because we didn't found differences in the capture, and we found differences actually between the sexes in the average height in the lab, but not across the time. But it's a thing that we want to explore a bit more to see if we see more results about it.
Uh, Also, are they social or mostly solitary?
Sorry.
Are they mostly social, the wood mice, or are they mostly
More
solitary
solitary,
animals?
mostly solitary actually. So
So
we maintain them in individual caging because of that actually, because they can be aggressive. For example, two males in the same cage probably would result in aggression patterns. So this is not what we want to have a more one more variable to add to the study. So we decided to keep them individually.
Uh, so what if you have like a bigger essentially cage? Do you think that's gonna be make it more? kind of similar to like natural conditions? Is that, have you planned of doing anything like that?
Yes, actually, this is one of the things that I'm going to plan is to increase the size of the cage and even decrease to see if the cages can affect, if the cages have some influence on it or it's is only enrichment because the space can have some influence on it. So it's a thing that we are thinking about doing. So experimenting different types of cages. So Mac 2, Mac 3, and Mac 4 to see what happens, especially on stereotypic patterns, to see if it reduces or not.
Another question from YouTube by Tang.
Mhm.
Why do you think the activity in the light darkness test decrease over time?
Actually, I think is the adjustment to the lab conditions, and because we saw this in our previous study in a barrier test, we saw the decrease of activity in the barrier test. So we conclude that probably is the adjustment to the lab conditions that lead the animals to make less and less activity along this time.
Um, Question from Abby. Can you comment a bit more on the sex differences in the urine marking? In the wild, do they show a similar level of variation?
So actually, the variation was different in the sex, but it doesn't seem to see enough informative. So what I think it's probably the sampling that we have may be enough to see other patterns, but maybe not enough to see the urine pattern. So we will need more animals to have a conclusion, to take a conclusion about the sex differences on the urine marking.
As we are nearing the end of the time, so you had a message for essentially take home message for people who are doing short term studies with this kind of mice. But do you have any suggestion on how to like keep the animals in the lab if someone is doing for long term experiment?
Actually, this is one thing that we want to do is to so I kept the animals for 30 days on the lab. So I want to extend a bit more this time to see if long-term studies can affect at the same time the at the same level the wildwood mice, because the we saw the adjustment that 30 days it's not enough to compromise their adaptation to habitat, but we don't know if a long-term study can affect their readaptation after release. So it's our next step is to increase the time of captivity to see how it goes.